Good evening to everyone. Welcome back to April webinar as a part of our series hosted as a part of the Center for Advanced Research in Brain Infections at CMC Vellore. So this project is actually a part of ICMR's funded initiative to look at how climate change impacts brain infections in India. And we're looking at various aspects of the project and how brain infections link to climate change itself. So as part of this, we have rolled out this series of educational initiatives where we have two flagship themes. One is the Let's Talk Brain Infections and Let's Talk Climate. So this webinar is the second of the series in the Let's Talk Brain Infections. This is co-hosted by AIIMS Delhi along with CMC Vellore, and we have expert panelists from NIMHANS Bengaluru. So it's my privilege to introduce the speakers for today. This is Dr. Divyani Garg, who's the Assistant Professor of Neurology at AIIMS Delhi. Her special interests include movement disorders and neuroinfections. And she is our proud alumnus from CMC Vellore, so we still share a very close relationship both ways. So happy to have you here, Dr. Divyani, to present to us on the neurology of rabies. We have Dr. Megha Brijwal, who's a clinician scientist at AIIMS New Delhi, who's primarily working with virology and microbiology in collaboration with mainly neuroinfections. She's got her main areas of expertise are post-transplant viral infections and arboviral diseases. And she'll be talking to us today on the diagnostics of rabies. And as senior moderators and panelists, we have Dr. Anita Mahadevan. Ma'am needs no introduction. She's a leading neuropathologist at NIMHANS Bengaluru, one of India's premier institutes, and she works in the pathology, and she also is a good, she's a clinician and a pathologist, we would say. Her main expertise is in the field of neuroinfections and also in the field of epilepsy. She's also been instrumental in setting up the brain banking and involved in the translational neuroscience research. And she has more than 300 publications indexed in national and international peer-reviewed journals. And we're very honored to have her as an esteemed moderator. We also have Dr. Reeta, who's a senior neurovirologist at NIMHANS Bengaluru. She is an expert at the neurotropic viral infections, and she's played a pivotal role in establishing the neurovirology diagnostics. And she's also brought up being the main collaborating center of excellence for rabies diagnostics at NIMHANS. So thank you, ma'am. We are honored to have both of you here to give us expert comments and lead the discussion and moderate these sessions. So over to Dr. Divyani.
Thank you, Angel, for a very kind introduction and for this particular endeavor, which I think is really very useful as a resource. So I'm a neurologist, so I will be speaking on the neurological aspects of rabies. And we know that rabies has been in the limelight for the last several months, in fact, actually. Especially, there was a lot of this came into the limelight when a 6-year-old girl sustained a dog bite, subsequently died of rabies in Delhi. And after that, there was a legislature to kind of take care of the problem of dogs, particularly the stray dogs. And ever since then, there has been a crackdown, and the animal rights activists have also been at loggerheads with this particular legislature. And but in the meantime, rabies continues to be reported frequently in the newspapers. And as of very recently, as of last month, end of last month, 26th of March to April, as you can see, rabies, there was a mass dog bite incident in a town of Assam. It's called Lanka. And a pet dog actually was bit around 40 plus individuals there, and then died 10 days later. And then the body was exhumed and rabies was proven. So as of this very month, we have this mass dog bite incident that has happened. Very recently, also, there was in UP a rabies scare when a buffalo whose milk was used to make raita that was served at a wedding subsequently died of rabies, and that triggered a huge panic, and 200 individuals rushed to get the rabies shot done. And that just is testimony to the fact that it's a dreadful disease. There is really, I would say, nearly 100% mortality, and that is the problem. It's a very rapidly progressive disorder, and we lose patients very quickly. So this is where we are as of now in terms of its impact. So I will be just going through this particular outline, and the diagnosis part will be covered by Dr. Megha. So I will not delve much into that aspect. So rabies virus actually is a Lyssa virus. There are many Lyssa viruses, and this is one of the Lyssa viruses, the genus under Rhabdoviridae. And it is a neurotropic virus. So 99% of rabies across the world, that is mainly Asia particularly, but also Europe and Africa, is because of transmission through dog bites. But in America particularly, bat bites are also known to produce rabies, and the specific feature of bat bite rabies is that because the Lyssa virus that is in the bats, it actually replicates in the epidermis and dermis, then even minor skin injuries can actually produce a full-blown picture. And worldwide, it is the children who are bearing the brunt of this particular problem. And it is labeled as a neglected tropical disease because I will show you also that it continues to be pervasive in countries where resources are limited, and some of the more developed countries have managed to eliminate this condition. India has the highest total number of deaths of human rabies that account for 35% of total global cases. But if you look at the highest per person death rates, then those are in sub-Saharan Africa. It is estimated, and I think, and it is widely acknowledged that this is in fact only it's a very small, I mean, it's an underreported statistic that we have, that there are 59,000 deaths annually due to rabies, and India reports around 20,000 deaths per year. But the majority of these deaths actually occur in Africa and Asia. And I would say that a large proportion of these are people who are living in the rural situation. They are underserved, and most of these individuals are children. So you could call it to be a neglected pediatric disorder, in fact. And this is just the areas in the world where rabies is confirmed to be present. And you can see that in the US and even in parts of Latin America and Europe, rabies has been actually eliminated. So they have achieved. Australia as well, rabies is no longer prevalent. And we can take some lesson from these Latin American countries in terms of the strategies that they have used to eliminate rabies, prior to which it has been a worldwide problem. So the transmission is through the saliva of an infected animal, which is either through a bite, open wound that comes in contact with the saliva, or an abrasion through conjunctiva or mucous membranes. And the predominant transmitters are the mesocarnivores. These are animals whose diet, 50 to 70% of their diet is carnivorous. And so dogs, jackals, skunks, all of these are in fact considered to be mesocarnivores, and these are the major transmitters, except for bats in America. And then there is a herbivorous animal called kudu, which is a type of a deer that has been reported to be a transmitter of rabies in Africa. Rarely, and unfortunately, it has also been reported through transplantation of organs. There was this particular report in the NEJM in 2013 where an individual, his organs, he died of a subarachnoid hemorrhage. His organs were transplanted to 4 other individuals who subsequently, in the next 1 month, succumbed to what was eventually proven to be rabies. And he had actually had a bite prior to he succumbing to the subarachnoid hemorrhage. So unfortunately, the transplanted individuals succumbed to rabies. And there is a assumed single case of perinatal transmission. So far, there is no case of human rabies resulting from the consumption of raw milk, raw meat or milk from a rabid animal. But of course, there is always a concern for that. So this is just a pictorial representation of the transmitters of rabies in various parts of the world. As you can see, in our country, it is the dogs dog bites, and that is why this whole hue and cry about stray street dogs that has been happening. But you can also see that there are skunks, raccoons, foxes, jackals, wolves, bats in various other parts of the world. So when we look at the pathophysiology, and just in brief, so whenever there is a bite, it is at the level of, particularly at the level of the muscle, the neuromuscular junction, the virus enters into the neuron through what are recognized several receptors. So the nicotinic acetylcholine receptors, P75, NTR, and NCAM. So the virus kind of replicates at the level of the muscle. It is taken up into at the neuromuscular junction into the neuron, and then through the dynein transmission, it actually traverses in this retrograde fashion towards the soma. Once it reaches the soma, there is a replication that happens over here. As you can see, new virus particles are produced and it continues to spread. So this continues still, as you can see over here, in a retrograde fashion. It eventually reaches the brain, and there it causes a neuronal dysfunction. Actually, it probably works at the level of the neurotransmitters, disrupting neurotransmitters, and causes a neuronal dysfunction. And then it starts to spread forward again to reach various other organs, the salivary glands, the eye, and then down the sensory nerves to the skin. And all of these are important because these fluids and these tissues can subsequently be sampled. It reaches these so that the life cycle can be maintained. It can be further propagated, presumably by contamination through saliva to another recipient. That is how the natural cycle would be. Somehow, the rabies virus manages to reach all the way to the brain without inciting much immunological reaction, which is why you see that patients are largely somewhat asymptomatic till they actually come in with florid neurological manifestations. And this is just to show you how complicated these mechanisms are through the various proteins that are part of the virus, like the NMGP protein, etc. They work on various aspects of the innate immunity and then the adaptive immunity, and somehow they suppress that while the virus is actually resting at the neuromuscular junction or traversing all the way up. And that is how nothing much happens clinically till it is actually in the central nervous system, mostly. So this is how the clinical staging has been described for rabies. So there is an incubation period which can vary from a few days to months to even up to more than 6 years. Then there is a prodromal phase which can last from a few days to up to a week. And then we come to the acute neurological signs, which is usually when the patients present to the medical facilities with either classic or non-classic rabies. And then there is an inexorable course to coma and death. And what determines at what time these various phases, what is the duration of these various phases, is determined probably by various factors like the host immune response, the age of the patient. So, for example, in children, it can occur faster. The virus strain, the species, the dosage of the virus inoculum, the site of the bite. So, for example, if it is more proximate to the brain, like in the hand or in the neck, then that traverses quickly, and so the incubation is less and the prodrome is less. And then the depth of the bite. So if it directly reaches the muscle or the nerve, if it reaches the bite, the virus reaches the nerve directly, then that will rapidly be transmitted upwards. So the incubation, as I said, is determined by a number of factors and can last from few days to as much as 6 years. So it's really important to kind of have this history in the background when we do take clinical history, to keep this in mind that the incubation period can actually be a little longer than we assume. The prodrome is the next phase, which usually occurs 2 to 10 days later. And this is characterized by non-specific symptoms like fever, headache, malaise, loss of appetite, flu-like symptoms. And then sometimes when the virus it reaches the dorsal root ganglion, it can cause a lot of sensory symptoms like tingling, numbness, burning, and pruritus. And this is a picture of somebody who had severe pruritus and scratched themselves. That would be on the limb of the injured limb or the site of the bite. That is where these symptoms might occur. So the acute neurological phase is the next one, and that is the one we are more familiar with. And this can actually present either in the form of what is called furious rabies or in the form of what is called paralytic rabies. So furious rabies is actually, it is so called because the patients are very hyperactive. There is a lot of agitation and confusion, and there is a lot of autonomic dysfunction that leads to profound frothing, hypersalivation, piloerection, and then there is a characteristic hydrophobia that happens to these patients, or aerophobia, and that is because they have a lot of pharyngeal spasms. So they become very fearful of any exposure of liquids to the throat because that would immediately create these severe inspiratory distressing spasms. And in the paralytic rabies, these patients come in less with all this agitation and central nervous system features, but they come in more with motor symptoms like quadriplegia, and that can be very progressive and then eventually leading to brainstem involvement with dysarthria, dysphagia, and this can actually, in fact, mimic more like a Guillain-Barré syndrome clinically. And then eventually, all of these patients, so in the paralytic rabies, the patient may actually be coherent and talking to you, but having profound motor weakness. But eventually, both of these phases will progress towards coma and death very quickly, as you can see. So when we talk about furious and paralytic rabies, the furious rabies is more common. It is seen in 67% of persons with rabies. And it is not really related to where the bite is or anything like that. Why somebody develops furious rabies and another person develops paralytic rabies is also not very is not very clear. But the deficits we have already spoken about. So if you have somebody who has an ascending pure motor weakness with areflexia predominantly involving proximal muscles, facial musculature, but the consciousness is very well preserved, then that would be something that would be like a paralytic rabies. When we look at the electrophysiological features, then these patients with paralytic rabies, actually they have evidence of peripheral demyelination or axonopathy. So they can actually, it can resemble to some extent GBS presentations. And then in patients who have furious rabies, that picture we are probably well aware of. So the weakness, and these patients can also develop a quadriplegia with areflexia, but that happens after they have become comatose and not before that. So yeah, some of the rarer manifestations that have been reported in the literature are a transverse myelitis, peripheral neuropathy, Adam-like presentation, ataxia, and acute psychosis. But eventually, this may be a confusion when the patient presents in the beginning, but the course of the illness eventually will give the clue where the patient very rapidly deteriorates, develops autonomic dysfunction and coma. These are some of the classical spasms in rabies that have been we must have all seen such patients as well, but this is nice video documentations of the same. So you can hear those spasms, and then these patients are offered water and they will resist it with a lot of fear. He is being offered water, and you can see how fearful, difficulty in accepting it. It's because water will trigger inspiratory spasms, and this is some
disorders due to rabies.
some of
One.
the patients from our country
developed rabies after being bitten by a stray dog. He is lucid and can raise his limbs on command. He has spontaneous, painful inspiratory spasm involving the pharyngeal, neck, and truncal muscles. Note the defensive posturing by upper limbs and the terrified look. Blowing air by a sheaf of papers triggers yet another spasm. Segment 2. 60-year farmer was attacked by a leopard and few weeks later developed rabies. He has claw and bite marks over his face, arms, and chest. Attempting to drink water from a glass and even bringing a glass close to him triggers frightening inspiratory spasms. Segment 3. 25-year man was bitten by a dog developed rabies, and survived. 1 year later, he is akinetic, mute, has generalized dystonia, and interacts only minimally.
So some of the survivors that we have, they get left with significant sequelae. And then in paralytic rabies, there is another feature that is called percussion myoedema. So when percussion is given over the muscles, this mounding or dimpling can be seen. That can be a clue. So those are the classical rabies, and then non-classical rabies are what is described in association with bat-related rabies. So here the sensory symptoms, neuropathic pain, radicular pain are more common. Sometimes in the bitten limb, patients can have immediately cuneiform movements during the prodrome. And then focal brainstem signs, myoclonus are more common, seizures are more common. This non-classical rabies is definitely less common, but it has also been reported with dog bite rabies. So generally, as you can understand from the clinical picture that we have seen, unless you have a history of a clear bite, which is really important to kind of dig into, the differential diagnosis can be actually very wide, ranging from our usual short duration central nervous system syndromes like autoimmune encephalitis, brainstem encephalitis, toxins, metabolics, electrolytes, drug use, related encephalopathy conditions, bacterial meningitis, encephalitis, viral encephalitis. The whole picture, if it is peripheral, then GBS and its mimics can all be a consideration. So investigations, I won't delve much into this, but if you look at the routine investigations, there may be hyponatremia, elevated muscle enzymes because of the spasms. CSF may be normal entirely, or it may show some pleocytosis with slightly elevated protein and normal sugar. If you do an EEG, that may just show generalized slowing. And then there is a serological test and other tests that Dr. Megha will take you through. Just radiology I will mention over here because that can be very useful if it is abnormal as a diagnosis. So CT is often normal, so MRI is preferred. So that's the imaging modality of choice. And what typically is involved is the brainstem brainstem and deep gray matter. Gray matter is involved more than the white matter. There is usually minimal enhancement and minimal hemorrhage unless it's quite extensive. And let me show you a few images. These are the classical images of rabies where you see that there is extensive and bilaterally symmetrical involvement of the basal ganglia, thalamus, and the brainstem. Sometimes the mesial temporal hyperintensities can also be there. So sometimes we think of autoimmune encephalitis in these conditions. Here, as you can see over here, there is this nearly like a putaminal hyperintensity predominantly. So these seem to be the areas of predilection for these patients, but also they can be this periventricular involvement that we see sometimes with NMOSD, in fact, very non-specific white matter changes, and then spinal cord involvement with myelitis may also be there. So we just had to look at the radiology without seeing the patient, then maybe the list of differentials would be really wide, like metabolic, toxin, etc. But so that is why it is important. But if you see these in the clinical context, and these are very, very classical of rabies. So management, actually, hospitalization is advocated so that there is less exposure to other people, and then the whole procedure shifts to palliative care. There are various models that are described, like the starfish palliative care model, where the whole focus is on actually sedating the patient, giving various benzodiazepines, analgesia, morphine, ketamine for this, and then agitation. Sometimes antipsychotics may also be used for the same reason. So the whole management aspects are focused on palliation. Now, there has been a lot of discussion in the past about the Milwaukee Protocol. So this was like a kind of considered a breakthrough when in NEGM in 2005, they reported one patient, a young girl, 15-year-old, who survived after sustaining bat-related rabies. And they actually produced this really stringent regimen that focused on supporting critical care and evading fatal dysautonomia while the body developed natural immune response, so that the Rabies could be cleared from the brain. So they did this by producing a therapeutic coma by administering GABA agonists and NMDA receptor agonists like imantadine, which also acted as antivirals, and a concurrent administration of these drugs was given. So the patient actually stayed for beyond a month and then survived with some sequelae. The Milwaukee Protocol has been beset with a lot of controversies. There's been a lot of criticism as to why to produce all this severe therapeutic coma when the evidence is not that clear, and there's a lot of risk associated with. People have come up with various modifications like the Brazilian protocol, etc. Really, it has been debated a lot, and as recently as December 2025, Dr. Jackson wrote about the demise of the Milwaukee Protocol for rabies. And then the main proponent of it gave a response saying that this seems to be a premature demise of the Milwaukee Protocol. And then there was another reply to Dr. Willoughby on the demise of the Milwaukee Protocol. So the argument being that it is not consistently shown to be associated with survival in rabies. So there are documented survivors, around 30 odd patients from various parts of the world without any clear protocol that had been employed by various people with the patients survive. The ones who survived, some of them have been left with significant sequelae. Now, if anyone knows this person, in 1885, it was Joseph Meister who was the first human to receive the Pasteur's live attenuated rabies vaccine on July 6th. And it is also an interesting tidbit that he went on to become a laboratory assistant with the Pasteur team. And then he was also the first human to receive the rabies vaccine booster when he underwent a second post-exposure prophylaxis when he was accidentally bitten by a guinea pig during one of the experiments. So this is Joseph Meister. The timeline of vaccine development, I'll just quickly finish this because of some time we have a little bit. And this is actually the mainstay of what rabies prevention is what we are actually focused on. So early concepts and the pre-vaccine ideas were all very superstitious. They would put dog hair onto the wounds, and there are lots of very interesting literature related to how rabies was handled before the pre-vaccines. And then investigators experimented with treating with attenuated saliva. And then from the Pasteur's era, we have had these live attenuated viruses derived from various sources. And subsequently, we have had better preservation methods, better protocols. The rabies immunoglobulin has come in. And then we are at this modern stage of cell culture vaccines. So these are purified, highly immunogenic with shorter vaccination schedules. So vaccination strategies are either pre-exposure prophylaxis, post-exposure prophylaxis. So pre-exposure prophylaxis is advocated routinely for our domestic animals, vets, animal rescuers, field workers, people who work in remote endemic settings where the dog bite incidents or vampire bat rabies are known to be present, and they are given pre-exposure prophylaxis. The advantage is that if you have a pre-exposure prophylaxis, it makes the injection of rabies immunoglobulin unnecessary after a bite wound, and post-exposure prophylaxis is after exposure. So in post-exposure prophylaxis, both are required depending upon the category of the wound. But if you have pre-exposure prophylaxis, then only the vaccine will do. So these are the WHO categories of wounds. Category 1, which is basically there is no exposure really, touching or feeding of animals, animal licks on intact skin. Here, washing thoroughly for 15 minutes with soap, water, and some disinfectant is advocated. No PEP is required. Category 2 is where there is nibbling of uncovered skin, minor scratches or abrasions without bleeding. So again, stringent wound washing and then immediate vaccination. Category 3 are the more severe exposures, single multiple bites, contamination of the mucous membrane or broken skin with saliva from animal licks, and then direct contact with bats. Then all 3 strategies, that is wound washing, immediate vaccination, and administration of the rabies immunoglobulin or monoclonal antibodies are advocated. So WHO retains its recommendation that the use of nerve tissue vaccines like Semple vaccine, etc., that were used earlier be discontinued, and they should now be replaced by vaccines based on rabies grown in cell culture or embryonated eggs. So evidence supports the use of either intradermal or intramuscular injection. So intradermal administration was advocated by WHO because it was studied to be cost-saving and also dose-sparing alternative. A systematic review has shown that the vaccines, when they are administered by the intradermal route for either PEP or PREP, they have efficacy that is equivalent to the intramuscular route. And for both of these, there are various sites. It's important to remember that it should not be injected in the gluteal region because it may not reach the site, and because there's a lot of adipose tissue in that area, so that is not recommended. So these are the sites for adults and children. And rabies immunoglobulin is to be given depending upon the category 2 or 3. Category 3 by passive immunization. It basically produces passive immunization by neutralizing the virus at the site of the wound before the immune system can respond. And this is to be injected into the wound, and this is the dose for the equine-derived ones, which are a little more hypersensitivity, maybe more with these products, but in resource-constrained settings, these are still an option. 40 international units per kg body weight, and if it's human-derived, then 20 international units per kg body weight. Skin testing before giving the equine rig is not to be done because it is an unreliable predictor of adverse events. And even if it is not available on the first visit, then it can be given up to 7 days after the first rabies vaccine administration. This is what I had shown you, and there are various regimens that are available depending upon. These are the various regimens that have been provided for the details, the routes of administration, and the days. And this is finally the One Health approach that has been advocated by a collaborative effort between WHO and other agencies like Food and Agriculture Organization, Global Alliance for Rabies Control, and they proposed the 0 by 30 strategic plan, which was to have 0 human deaths by rabies by 2030. So there has also been a kind of a push in India to think about where we are in 2030. They have also said that mass vaccination of dogs is actually the principal strategy for interrupting rabies transmission between dogs and reducing transmission to humans and other mammals. And this is the global framework that they have advocated, which is focused on the 5 pillars. Top R, S is for socio-cultural, T is for technical aspects related to vaccination, O is for organization, leadership, partnership, coordination. Political is P, and then resources. So it's a very elaborate document that has been provided as for the strategic vision. I'll just finish by reminding you all that 28th September is actually World Rabies Day. It is the anniversary of Louis Pasteur's death, and he was the person who developed the first rabies vaccine. Yeah, thank you.
So, thank you, Dr. Divyani, for that comprehensive lecture. So, may I invite some inputs from Anita ma'am on the topic?
Thank you, Dr. Angel, for the invitation. It's been a very exciting being on this because Dr. Divyani gave a very beautiful, comprehensive overview of the clinical and the current state of vaccination recommendations and a hope that by 2030 will be free. I think as a pathologist, what has always fascinated me about this virus is it's a master. It's a master which has, I think, very successfully, it knows our immune system, it knows our physiology, it knows our anatomy. It's the best neuroscientist, I think. So like Divyani said, we know it's 100% fatal, right? But we still don't know what causes death. She mentioned that it's a very variable incubation period. We still don't know why it is so variable. She mentioned about the dramatic clinical picture of hydrophobia, aerophobia. We still don't know why. And interestingly, dogs, animals, lab animals don't develop hydrophobia, aerophobia. It's exclusively human. So if you look at the virus per se, it's very unique entry intra-axonally. We know about all of that. But what it translates into is all of what you see clinically. One, it never has a vireme, which tells you why the immune system is never exposed to it. So what does that mean? You can't do a PCR, you can't do antibody studies. And when it enters into the brain, what we have seen at autopsy, we have 40 plus autopsies that we have of rabies. And the most intriguing thing is, unlike every other viral infection, there is minimal to absent inflammation, which explains why you don't see cerebral edema, which explains why the MR changes that Dr. Divyani had described is usually seen only in the terminal phase when the blood-brain barrier opens. Until then, it's happy inside. And because it is exclusively intracellular, we are not seeing an immune response at all. So we try to look at whether the incubation period has any explanation other than what everybody talks about. The proximity of vibes, the dose, the inoculum, the viral strains, etc. So we just looked at anatomical regions, the entire brain regions that were involved by the virus. And very intriguingly, one explanation that we could find was, in those with very short, ultra-short incubation period, which is in weeks, the critical areas get affected first, which is your midline raphe, the thalamus, the brainstem. Rest of the brain is essentially not involved. But if you look at long incubation from months to years, the entire brain will show you viral antigen. So if it cleverly localizes to only critical areas, that is what seems to be causing the very short, ultra short incubation period. So it's also probably dictated by the route it chooses when it enters into the brain. Of course, it could be viral strains too, and maybe Dr. Reeta can comment because she's done a full-length sequencing of the viral genome and looked at viruses. But one other thing that we saw at autopsy was every viral infection, I think apoptosis was a catchword for a long time. So we looked at whether rabies causes apoptosis, and very interestingly, it doesn't, which makes sense because it has to keep the neuron within which it is living, it has to be alive. If that undergoes apoptosis, it's going to die. But what does undergo apoptosis, very interestingly, is all the inflammatory cells that come into the brain. So whatever inflammatory cells, it causes apoptosis and removes it. So it continues to survive. So these are a couple of things that kind of came out from autopsy studies. The cause of death, we tried hard to find because there is absolutely no cerebral bleed. Even post-mortem, we never see terminal. No edema. It's a very beautiful brain. You can use it to teach anatomy. But fact is 100% fatal. So one of the studies that Dr. Reeta herself has done is to look at mitochondrial dysfunction. So maybe we can invite Dr. Reeta to give comments on what she found at with autopsy material. Dr. Reeta?
Yes. Yeah, I'll be brief probably because we are short of time. So thank you very much, Anita, for describing that wonderful pathology seen in rabies. Also, thanks, Dr. Angel, for this invitation and Dr. Divyani for a very comprehensive talk. So yes, we did study, one of my PhD students studied whether there is mitochondrial dysfunction, and we studied it in brains, both human and animal brains, lab confirmed with rabies. And yes, there's not much time to explain in details, but I would just like to say, yes, we did find some evidence of mitochondrial dysfunction, which might be leading to the neuronal dysfunction, which we see in rabies. I'd also like to say three more points in brief. So Dr. Divyani, you very clearly mentioned about RIG and monoclonal antibodies. I'd just like to add that HRIG is not manufactured indigenously. It is imported and therefore it's very costly, whereas equine RIG is manufactured in India. And we have two monoclonal antibody products now in India. You mentioned one, a single monoclonal antibody product by Serum Institute of India. And there's one more where it's a cocktail of two monoclonal antibodies, that is Twin Rab by Zydus. So both are good products. One other point you mentioned about pre-exposure prophylaxis. So that is one thing that as doctors, all of us need to advise everybody that we do not need to wait for a dog to bite us and then take a PEP. We can always, all of us can take PrEP, especially children who are vulnerable to dog bites. So pre-exposure is what we should be advocating because that is something pediatricians should advise, because the IAP calls the rabies vaccine as an optional vaccine in India, and people can take it as one of the cheapest vaccines available. And the last point, we have recently done a study because see there has been a lot of news whether there are vaccine failures which are leading to rabies despite PEP. But we have published a very good study recently in CID where we found that most of the so-called breakthrough infections were because of deviations in the PEP, mostly because of healthcare lapses, or in some cases lapses from the bite victims themselves. So this is again a very important point for us in the medical community to remember, that we really need to know the protocols very well if we are going to treat any bite victims. Over. Thank you, ma'am. Thank you, Dr. Anita and Dr. Reeta. That was very educational. Now may I invite Dr. Megha to give her talk on updates in the diagnostics of rabies. Over to Dr. Megha.
Hi, is my slide visible?
Yes, yes.
Yeah, thank you. Right, so I'll be talking about diagnostics in rabies, and I think Dr. Divyani has made my job quite easy. She has described beautifully pathogenesis of dengue, and that's where we are deriving what are the tests that we are going to employ for laboratory confirmation. So just for a virology, this is a bullet-shaped virus about it's a small virus which has an RNA genome, and its surface is consists of these G protein, which are peplomers on its surface. It's a very crucial antigen, which is a virulence factor, as well as the protein which is involved in immune evasion, and virus neutralizing antibody would actually, when you administer vaccine, would be induced against these proteins. So knowing this title is also important in neutralization assay if we follow through. So in laboratory diagnosis, broadly, we cover rabies diagnosis as an ante-mortem or a post-mortem diagnosis. So different samples. Many a times we are not successful in getting a post-mortem sample where autopsy facilities are not available. But in lot of cases, even ante-mortem samples are missed where there is clinical suspicion is low. Another aspect is after a bite of an animal, observing the animal and whether if the animal is already symptomatic or rabid, whether sacrificing that animal and getting the sample from the animal is possible. So ante-mortem samples, different samples consist of skin biopsy, which is mainly the nape of the neck just below the hairline, which has those proximal region, which consists of the hair follicle, where you can see the nerve innervation. So we are looking at innervated tissues. Samples could also be corneal impression smears, CSF, saliva, and blood. Postmortem samples are usually brain and spinal cord. So we'll be talking about laboratory diagnosis in following headings. First, discussing about the staining, then antigen detection, molecular assay, antibody detection, and further, there are a few newer things in rabies diagnosis. So as ma'am had just very nicely described, very well-preserved brain, but certain regions of brain, including brainstem, cerebellum, and other areas of brain, needs to be prepared for diagnostic purposes. So first thing in pathology, of course, is histopathology and staining. So we obviously know about Negri bodies, which are nothing but rabies inclusion bodies. Inclusion bodies are the areas where there are particles of virions which are aggregated. These are intracytoplasmic, as we can see in the figure. They are actually outside the nucleus, and these are small, about 2 to 10 microns, very sharply demarcated. The distinct color, which is described in cellular staining as cherry red color, is seen. And it actually, if you see very carefully, there are small stippling insiders, which are dark blue granules, which are seen inside. So you can see these Negri bodies in brain tissue as well as in the impression smears. If you prepared a brain impression smears, cellular stain is nothing but a combination of basic fishine and methylene blue. And common sites where do you see these typical bodies are hippocampus, mainly the amenson, the cerebellum, and other areas depending on where the virus is replicating. Few other stains like Mekenfold, Man, Jimsa has also been used. Further staining, histochemistry actually adds on to the diagnostics, where you can actually look at rabies virus antigen in these formalin-fixed tissues. These processes involve detection of antigen using specific anti-rabies antibodies, be it monoclonal or polyclonal, which makes the identification much more sensitive and specific. So another test, which makes it highly sensitive and specific, is a fluorescent antibody test. These are WHO recommended tests, very widely used, and especially preferred in animal tissues when we get. And here also, you can treat it with anti-rabies serum in a single step using monoclonal antibody, and a double step using a anti-antibody, a globulin, which is conjugated with a fluorescent dye like FITC, fluorescein isothiocyanate. It's mentioned that its sensitivity reaches up to 95 to 99% if you're using fresh specimens. Usually the results are available within 2 to 4 hours. The staining and visualization would probably be about 30 minutes. while acetone fixation of the sample may add on to some other time. So it makes it much more rapid in special circumstances. So as we just mentioned, can be done in single step or double step. So it can be a direct fluorescent antibody test, which is DFA, and some people call it DFAT. And other, it could be an indirect way using an immunofluorescent assay. Samples, again, here could be directly taking impression smears, whether it's brain or corneal impression smears, or even tissue samples like brain or skin biopsy can also be utilized. DFAT has now become standard for routine diagnosis, especially when animal brain samples are received in the laboratory. Adding on to this, a quite similar test is something called DRIT, Direct Rapid Immunohistochemical Test, which is a rapid immunohistochemistry staining with anti-rabies antibody. The only difference is rather than using a fluorescent antibody, here another chromogenic marker with immunoperoxidase maybe is tagged. Now the mainstay of diagnosis focuses on RT-PCR or molecular-based testing. Since it's an RNA virus, it's a reverse transcription PCR. Different samples like tissue, homogenates, skin biopsies, saliva, CSF can be tried. Samples here could be antemortem or postmortem. The idea for PCR is targeting a highly conserved region in the viral genome. So most often the target genes in PCRs tend to be N gene, which is a nucleoprotein gene, and the 3' leader region, which is just in the 3' region to the N gene. It has the added advantage of being very rapid. It provides high sensitivity, and especially when the DFA results are inconclusive or indeterminate. PCR can also be supplementary in those conditions. Now, there are different modifications of PCR which have come up over a period of time. Traditionally, we had these gel-based PCR where we look at the bands at a specific amplicon size. Following this, you had nested RT-PCR, which provided increased sensitivity. Nowadays, the mainstays are real-time RT-PCRs, where a fluorescent dyed probe is added in addition to the primers, and you have real-time amplification, which can be visualized. You can also combine a lot of target together, which is called multiplexing. So you can have multiplex real-time RT-PCR looking at the quality of the sample and the target of the rabies virus gene in a single reaction. So just to give an example, there are a lot of standardized validated assays available. So one of these protocol is something called LN34 assay, where the LN is the leader N gene and 34 was the set of primer where it was designed. You've had different variations of these LN34, and this is like one of the recent papers where they actually modified this protocol, improving the primer performances, and they multiplex this format. So a lot of standardized assays are available, which labs can actually adapt and use in diagnostics. So just to add on, not just in-house, there are also commercially available PCR assays available, and Indian manufacturers are also now producing these kits. These are few of the examples. One of the example here, TrueNat, is actually a single sample cartridge-based test. It's very widely have been used in TB diagnostic as well as in COVID-19 diagnostics. And there are other targets now. There are a lot of kits now which are available for IVD as in vitro diagnostic. But again, for an individual lab, a slight validation and quality performance for these kits is something which is essential. So now moving on to serological or antibody-based assays. So first looking at whether the neutralizing ability of the antibodies can be measured. There are few popular tests like RFFIT, Rapid Fluorescent Focus Inhibition Test, and Fluorescent Antibody Virus Neutralization Test, which are available. But again, in antibodies assay, we have to understand that it cannot differentiate between an antibody response to vaccination or it is a result of infection, because now many of people who are coming into hospital might have a history of receiving vaccination, which could be as a PrEP or maybe a recent PEP, where we are talking about those breakthrough infection cases. Also, if you detect a neutralizing antibodies in CSF or serum of an unvaccinated individual, that would be one which would be confirmed as a positive test for rabies virus infection by these serological assays. A serological titer of 0.5 or more is considered evidence of an adequate immune response if you're following it up as a post-vaccination response. So again, RFFIT, again, this test is generally done in cell cultures. So it's not able to adapt it very easily adaptable in a lab for routine testing. So there are easier formats of serological assays which are available, like ELISAs, modified CIP, or indirect immunofluorescence assay, which detects antibody, where you can actually now, depending on your detector isotype, you can have IgM or IgG antibodies also, which are measurable. Just to introduce here, while ELISA being the most popular method, now there are also rapid assay and ICTs which have now come into the market, but they are not available for human use. ICTs are nothing but immunochromatographic tests, which are rapid assays, which are results is usually available in within 30 minutes. So there is generally in the diagram, as we can see, there is a conjugate pad, and you can load the sample here. The antigen in the sample can migrate further, and you can have anti-N monoclonal antibody at the target site, which will capture these antigen. So you can have rapid assays which detect antigen. So samples could be a cell culture or a tissue homogenate, which can be utilized here. So this is one of the example of commercial assay, which is available, but only for veterinary use. And it uses canine and feline saliva samples, and it claims to provide a sensitivity of 85% and specificity of almost 100%. So, and they advise it as a screening tool with an approximate cost of about 250 to 300. Again, these as an IVD test for human use, these have not been validated. There are few ICTs which are in research mode for antibody testing in human samples. So just to complete, there are isolation. These are again not part of routine laboratory diagnostics, but as we also mentioned, now there are cell culture-based vaccines, which are the preferred vaccines, which are available. So it is a culturable virus in murine cell lines and hamster cell lines, animal inoculation, embryonated egg. If we remember, the earlier vaccines which were present were from these methods, and now the preferred are the cell culture-based vaccines. So of research interest, electron microscopic view, we can see this beautiful bullet-shaped virus. So just to summarize, you have different formats of laboratory detections with antigen detection, molecular base, where you're detecting the RNA, isolations, or antibody detection. Of these, the most popular test recommended by WHO as well as by the Animal Health World Organization are the fluorescent antibody test, the DRIT test, and the molecular PCR-based test. So now just to add on, are we looking for immune response to rabies vaccine? If somebody has been given a PEP, it is not recommended routinely to check for seroconversion. But in special circumstances, this may be required, especially if the person is an immunosuppressed person. And as I think Madam has earlier mentioned, if there is a deviation from prophylaxis schedule, because these are the groups where breakthrough infection, or where we are terming it as vaccine failures, could be a possibility. Another indication which has been mentioned is when pre-exposure prophylaxis is given for high-risk group, like laboratory staff or clinicians or veterinarians who are handling this, or even travelers from rabies-free areas which are traveling to endemic areas. There, this neutralizing antibody titer can be measured every 6 months for up to 2 years, and then every 2 years. And booster recommendation is also there if the titer falls below less than 0.5. So just to complete, we talked about antemortem diagnosis. So we have this US CDC data from 1990 to 2024, which talked about 4 recommended sample types like saliva, nuchal skin, serum, and CSF. And they mentioned 4 antemortem sample types. Wherever it was available, a combined diagnostic sensitivity was about 100%, and risk of false negative results were less than 2%. But in cases where the sample type were less, the sensitivity got compromised, and where only one sample type was available, the sensitivity was less than 65%. This probability of detecting this virus generally favors in term when the patient has presented. So saliva, nuchal skin samples had higher positivity in early illness, while antibody, of course, was higher in serum and CSF in later in the illness. So what it actually tells us is actually a multimodal, multi-sample approach when we are looking for rabies diagnostics. Of course, now There is metagenomic NGS, which has started to become integrated in some of the lab, especially in Western countries. In India, again, it's at infancy, but people are in research mode talking about viral metagenomic NGS. This is different from bacterial or parasitic NGS because they tend to have a single gene, which is a consensus common gene among those forms. But in viral, you can only have a targeted approach. You suspect rabies, so you test for rabies. Now it is time to look for untargeted approach or an unbiased approach, which can be done through a viral metagenomic NGS approach. You can have an untargeted metagenomic NGS, and you can also add on a probe capture approach where you can design multiple probes against different targets. Sample utilized here have been CSF and saliva. So this is again only on research mode, and you have several case reports where utility of metagenomic NGS has been shown, but it is limited by the high cost of this method as of now. It is resource intensive. You require specific laboratory infrastructure and bioinformatics expertise by the lab. But it does have some advantages, especially in atypical cases, paralytic cases, and especially where concealed or unclear exposure history is there. So just to give an example, this is a case report from China where a case of paralytic rabies was identified by metagenomic NGS. The authors further did a review of literature, and they found further group of 5 cases here, which were initially identified by NGS, and another group of 3, which also confirmed diagnosis by metagenomic NGS. So at the end, I would also like to mention here, we have talked about PEP. We are not going to go into that, but if we see this box here, if the recommendation is from area which is non-endemic, like US, they do mention animal testing first, and if the results are available within 24 to 72 hours, then to start PEP. But this does not follow through in our area because of high endemicity. But testing and a good turnaround time is something which can make testing and then starting PEP feasible only in non-endemic areas. National Rabies Control Program, of course, mentioned different aspects, and of these, surveillance system strengthening and strengthening of regional laboratories under NRCP for rabies diagnosis has also been emphasized. So just to summarize, antemortem diagnosis does remain a challenge in most of the endemic countries. This underdetection is likely due to undertesting. And of course, timely diagnosis is critical to clinical and public health decision making, and high index of clinical suspicion is required, especially in unexplained acute progressive encephalitis or in paralytic diseases. Of course, our approach should be a utilization of multimodal method, including molecular testing, and metagenomic NGS may have some utility in future for these atypical neurotropic infection. And at the end, I would definitely like to say that this theme, Act Now, You, Me, and Community, is a together effort, and One Health definitely plays a huge role in rabies. Thank you.
Thank you, Dr. Megha. That was a fantastic update in the diagnostics. May I invite Dr. Anita and Reeta ma'am's comments on diagnostic aspects as well.
Yeah, Dr. Megha, very nice review of antemortem diagnostics. I think from my side, I just want to say one thing, which is probably not going to be very popular. The gold standard would remain tissue diagnostics. So of course, antemortem diagnosis plays a very important role. It has enormous challenges, but please keep in mind that tissue diagnosis is the gold standard. So even if you cannot get a complete autopsy, a simple transnasal brain biopsy can be done in the bedside itself so that you can confirm a diagnosis of rabies. I think if somebody is interested, I can share how it is to be done. But I would probably invite Dr. Reeta to share her very vast experience of antemortem diagnostics. Reeta? Dr. Reeta, it's over.
Yeah, thank you, Anita. Dr. Megha
wonderful presentation. I think you have covered most of it. So I would also like to add to what Anita said about brain diagnosis. So the gold standard in rabies remains testing of brain. And that is the main drawback in India, that we as physicians do not convince the family members for an autopsy. And like Anita said, even if you cannot do an autopsy, you can just take an intranasal biopsy that Anita is extremely skilled at. In fact, we had a case in Ghana where Dr. Anita, through a telecall, she guided them to take a biopsy through the nose, and that tested positive. So all of you also, wherever there is a need, please don't hesitate to contact Dr. Anita, and I'm sure she'll be very happy to guide you to take an intranasal biopsy. Other than that, antemortem diagnosis, yes, you have rightly said, Dr. Megha, it continues to be challenging because the overall sensitivity is quite low. So you need to test multiple samples and serial samples to increase the sensitivity. So at NIMHANS, we get samples from the entire country, and our positivity rate for antemortem samples has been just around 40 to 50%. Now, that is because we don't get all samples from all the patients. And there are so many factors, that the transport conditions and the quality of the sample, which also impact the sensitivity. Therefore, the brain sample is the best sample to rule in or rule out rabies, because remember, antemortem tests can only rule in a diagnosis of rabies. If all the tests on antemortem samples are negative also, you cannot rule out a diagnosis of rabies. So ruling out can be done only on brain tissue. So this is something also, as a community of medical professionals, we all should remember. Yeah.
So we'll take a couple of questions for discussion. So we have, so the bite from a person or coming in contact with the saliva that are affected with rabies, can it transfer rabies in some cases? So this also brings us, like, anytime we have a positive PCR in ICU, everyone goes into a panic mode. Till then, even a couple of weeks back, we've had a patient where, in fact, we managed as a GBS, no exposure history, according to the relatives. And this gentleman came with severe bulbar, had to be intubated, and we even started Plex. So it's a good question. How do we go about it? Any comments from the panelists? Ma'am, Dr. Divyani.
The bite from a person.
Yeah, or coming in contact with their saliva. So
I mean, I think just saliva exposure, theoretically, I know, but it is not enough. A bite from a person, it is like, I mean, that would cause a breach of the skin. And therefore, if you were to go by the category of the wound, then that would constitute a need for a post-exposure prophylaxis. But so far, person-to-person transmission, like by this route, has not been reported yet. So I just want to say that.
Yeah, can I also add on to what Dr. Divyani
Yes, ma'am.
said?
Yes, ma'am.
Yeah, so she has rightly said there is no reported human-to-human transmission case except through organ or corneal transplants. But having said this, theoretically, all biological fluids, including saliva, urine, tears, all these can carry the virus. And therefore, the theoretical risk of rabies always exists, especially see when you're intubating a patient, and generally such patients hypersalivate. And if there's a splash of saliva on mucous membranes of the eyes or the oral cavity, there is always a risk. The second thing to consider also is the anxiety and the phobia in healthcare workers. So in such cases, I always say in rabies, it is always better to err on the side of caution. Like, give them the vaccine. It is not going to cause any harm. And as you said in your presentation, it's going to protect them lifelong. They don't need
any rabies immunoglobulin, anytime for any future exposure, they just have to take 2 boosters, day 0 and 3, on any subsequent exposure throughout the rest of their lives. In fact, Dr. Anita's entire team, even the team which works in mortuary, they do a lot of postmortems. They may never know whether it is a case of rabies. So we routinely vaccinate all of them. In fact, whoever comes in our laboratory or Dr. Anita's department, they all get a pre-exposure rabies vaccine.
I think the key thing is that there is no harm.
Yeah.
And the benefit is enormous.
So next question is the dorsal root ganglion reservoir site, and are there any milder variants in the people with pre-exposure prophylaxis were noted?
I think dorsal root ganglion per se is not a reservoir site. It is definitely an important conduit when the virus retrograde in a retrograde way goes from the neuromuscular junction to the peripheral nerve, to the dorsal root ganglion, and to the spinal cord. It may temporarily stay there while incubating, but it's not really a reservoir site. I think ma'am, Anita ma'am can also add to that. And I don't understand the second question. Any milder variants in the people with pre-exposure prophylaxis, milder variants of rabies?
Yeah, so I don't know whether they mean some endemic titers. Is that what the question is? Possibly.
So I think with respect to dorsal root ganglia, I agree with what Divyani said. But just an intriguing study that we did, I was very taken by the fact that, symptomatically as well as imaging wise, it is so HSV-like. There is medial temporal involvement, there is limbic involvement. So I took out trigeminal ganglia from patients who died of rabies and looked at whether there is viral antigen within that. And to my surprise, it was full of viral antigen. It tells you that distribution is explained by trigeminal involvement, but whether it remains latent there, gets reactivated, is something that is difficult to answer. So probably what we'll have to do is animal inoculation studies, and before symptoms set in, if it goes into the trigeminal, then we can probably say that, yeah, it is a site for viral proteins. But it's a very good question. It is something that I think is intuitively probably makes sense. So maybe Dr. Reeta, can you come in with any of the genomic sequencing results that you had done?
Yeah, so if it's about variants, so in India, we don't see too many variants. It is like Dr. Divyani mentioned that there are many Lissa viruses. So there are 17 different species of Lissa viruses which can cause a potentially rabies-like illness. Now, in India, we assume that we have only one virus, which is also called as the rabies virus, the first prototype virus. So we have not seen or reported any other Lissa viruses, including bat Lissa viruses, are also not reported from India. So we have the dog strain, which is predominantly seen. In fact, whatever we get from wildlife also is the dog strain.
Sure. So next question is, even though effective vaccines are available, rabies still persists. What are the biggest system level failures preventing the achievement of WHO 0 by 2030 goal? Or is it because of any genetic variations in the viral strains that influence the vaccine efficacy or diagnostic sensitivity?
So this I'll answer because this is a misconception which I have heard from all doctors, and I mean rightly so, because that is what we see. So there are two, as I said, we have done several studies where we have found that vaccine efficacy is not a problem, except probably a few cases where the cold chain was not maintained or some counterfeit vaccines were circulating. But otherwise, this vaccine is a killed vaccine. And therefore, the potency is not very easily impacted. Now, coming to the viral strains, because of COVID, now everybody knows a little bit of genomic sequencing, and they feel like COVID, we have multiple strains of rabies virus.
No, though it is a RNA virus like the COVID virus, relatively it is a very stable virus. We have done extensive genomic sequencing. In fact, we have published close to 700 full-length genomes of the rabies virus over years. And we have found that there are no significant mutations in the areas which can contribute to failure in vaccine efficacy or even the HRIG or monoclonal antibodies. So yes, of course, we need more data, but as of now, I can quite comfortably say that most of the PEP failures are because of deviations. The wound is not washed. The vaccine is not given properly. It is given in the gluteal region. Immunoglobulins are not given at the local site. They're only given intramuscularly, which is of absolutely no use. WHO now clearly says give only local infiltration. It is okay if you don't give a systemic dose. Suturing the wound without giving rig first. Generally, suturing should be avoided in potentially rapid wounds. So a lot of deviations we have seen, even dosages. We've seen pediatric patients getting half the dose of a rabies vaccine. We must know that the dose and the schedules are same for a newborn, immunocompromised, elderly, everybody. The schedule and the dose is the same. So there are a lot of misconceptions even among the medical community. So that is what we need to target. And coming to this question, like what is stopping us from getting freedom from rabies? I would say that every bite victim should get PEP, which is anyway free at most government centers, but it should be timely, adequate, and appropriate. But just taking PEP is not going to eliminate rabies, because if we don't take care of the main source, which are stray dogs, then it is like mopping the floor with the tap on. So yes, parallelly, we have to focus on the source, as WHO rightly says. Elimination of rabies in dogs, which is mostly through mass dog vaccination, is what will take us to that goal. And there are pockets in India, like the state of Goa, who have managed to do that. And it is doable, though it is a formidable task. Yeah, thanks.
Okay, ma'am. So, why host immune response fails post CNS entry despite vaccine-induced antibodies? As I presume, if you have a case of vaccine failure, I presume that's what. Like, so, ma'am, one more phenomenon, this early death phenomenon. I wanted to discuss that from our side. Like, they say it's a race between the virus and the vaccine. But sometimes when the virus reaches the CNS before the vaccine response kicks in, moment the vaccine response boosts, that can actually add to the viral advantage. So, do you have any experience with this early death syndrome, where it's a competition between the immune response and the vaccine, and the viral action? Do you have any experience with that, ma'am?
Yes, so this is not the early, I wouldn't call it early death. I would call these cases as true PEP failures.
Okay.
So remember, the vaccine can protect somebody only before the virus enters the nerves. The antibodies can neutralize the virus at the site of the bite before it enters the nerves. Once it enters the nerves, then even God cannot save that person. So now we see a few cases of true PEP failures. Now that is where what you mentioned. Now say suppose if it's a child, there are multiple deep bites on highly innervated sites. Like Dr. Divyani mentioned, if the bite is directly inoculating the virus into a nerve, no amount of immunoglobulin or vaccine is anyway going to protect that bite victim. And these are cases where despite PEP, we see deaths, and that usually we see the incubation periods are very short also in such cases. So these are very rare, but yes, we do see these are true cases of PEP failure where everything is given on time, but most of these cases, even before the end of the month, before they can take the last dose, they develop rabies.
So, two final questions. So, does a patient require vaccination and immunoglobulin in a case of grade 2 or grade 3 bite by a dog previously vaccinated for rabies? And at what stage post-exposure does a nasal biopsy become reliably positive in relation to symptom onset? And can this be standardized in tier 2 or 3 centers? So, answering whether it's a, yeah.
Anybody else wants to answer? Otherwise, I'll answer.
Go ahead.
Anita mentioned about the transnasal brain biopsy, not nasal biopsy. I think Anita ma'am can sort of put a bit of light on it.
Yeah.
Yeah, so it's difficult to give an absolute timeline, but this is about the olfactory you're asking, no? Somebody was asking.
They've
So
written nasal.
orbitofrontal is the area that gets targeted when you do a transnasal biopsy. Now, since that is part of the limbic system, and limbic involvement is kind of prevalent in most cases, so targeting that, the sensitivity specificity is very, very high. There are other sites also that you can target. You can go through the foramen magnum and get the cerebellar tissue and the brainstem, which you can get brainstem 100% positive. But how many other transnasal we have done, we've never seen a negative test. So having said that, I think it is involved, but I can't tell how early on. Most of the localization of rabies viral antigen need not cause symptoms. So by symptomatology, you won't be able to say this area is involved, that area is involved. It is a silent kill, simply because it doesn't generate inflammatory cells. Only if it does, then that causes symptoms.
Yeah, I would say, see, we are talking about post-mortem transnasal biopsy of the brain. Remember, we are not talking about ante-mortem brain biopsies, which is not recommended for diagnosis. So when it is post-mortem, at that phase, even Anita would agree, the entire brain is studded with the virus in humans. And we generally nowadays, we do a real-time PCR on a brain tissue. Unlike probably a histopathology which might require more sections to be screened, a PCR is a highly sensitive test, and even a small piece from any part of the brain would still give a positive. So I don't think the timeline or the area matters in such a situation, because it's always post-mortem brain biopsy that we are talking about.
And if you're bitten by a vaccinated dog, do you still need a vaccine? So if it's a category 2 or 3 bite, that's the last question.
So see, rabies immunoglobulin along with vaccine is recommended in category 3 patients, category 3 exposures, and category 2 exposures in immunocompromised patients. Remember that. Okay, so this is where we need it. Now, this is a very common question that we get, that it was my pet dog which bit me and I have vaccinated the pet dog. So Dr. Megha also showed us that in non-endemic countries, they would observe the animal for 10 days or sacrifice it and test the brain and then take a PEP decision. In a country like India, we should never ever do that mistake. So whoever comes and tells us my pet dog bit me, we don't really trust them or believe them. We just say, start your PEP. At the most, by day 10, if the dog or the cat, and remember this observation period applies only to dogs and cats, not other animals. So if the pet is alive after 10 days, then you can skip the last 2 doses of PEP, which falls on day 14 and day 28. But otherwise, please do not delay initiation of PEP just because it's a pet dog or a pet cat. Because even if they are vaccinated, we have seen in many animals, the antibody titers are not adequate because there are so many reasons. The quality control of animal vaccines in India is not regulated as well as human vaccines. Then any malnutrition, helminthic infections in animals can lower the immune response. So just evidence of vaccination is not complete evidence that the animal is protected against rabies. So there are lot of caveats, ifs and buts. So it is always better to just go ahead and recommend post-exposure prophylaxis.
So I think, yeah, so that brings us to the end of the webinar. I think we had a very good discussion, and it was a very, very informative and useful. And we'll probably be using this lecture also in our project channel, so that even if people who didn't have the opportunity to attend this can watch it and learn from this later. So I extend my heartfelt gratitude to Dr. Anita and Dr. Reeta for sharing your experience. And it was every time we hear from you, it is, it's very inspirational and very nice. Thank you, ma'am, for being here. And special thanks to Dr. Divyani and Dr. Megha. Both of you, your presentations were well applauded in the comment session. Also, everyone said your presentations were clear and very informative. So thank you so much, and we'll meet in the next webinar, which will be on climate, which on focusing on the health heat impacts and a little bit of input into the Ahmedabad heat action plan, which is planned for the next week. So I think there's probably one more. I think somebody wanted to ask another question. Yeah. So. Okay, probably not. Okay, thank you. Thank you all. Have a nice evening.
Thank you.
Thank you.